means you usually give them
Looks
funding
like
and resources and they want to carry.
Hey, all.
Hello.
Thank you all for joining us today, and thank you, Ben and Michael, for switching your presentation time.
Anything for you, Ellen?
And you know there'll be a cost later, right? That's how this works. You know that, right? Don't fall for that story about anything for you. It's turnaround. You know how that goes.
Hi, everyone. It's Donna Robertson.
Hey, Donna.
Here's Mike's on now. Kyle's on now.
Hey, yep, I'm here. All right. A couple people still logging on, and then I think we had talked. Ben, were you going to be good to do introductions today?
I'm actually starting the talk, and then I'll shift it over to Michael.
Perfect. Okay, so for our for today's edition of our Alzheimer's disease ideas forum, we had a couple of folks here who were very, very kind to be able to fill in for somebody who had canceled, and they were supposed to talk in a couple of months. So we're going to hear from them today, and I'm super enthusiastic about it. I know I reached out to a few other people as well. I see Scott's on, Scott Floyd. He's a radiation oncologist who's interested in, Scott, in sort of neurosphere, three-dimensional organoid cultures of things. And so anyways, we're really excited to hear from Dr. Ben Barr and from Michael about their work at UNC Pembroke in this regard. So I'll turn it over to you, Ben.
All right, I'm going to show some slides. Michael Almeida is my lab manager and graduate student working at UNC Pembroke. And the key point of my laboratory is looking at brain vulnerabilities that can influence how we become susceptible to dementia. We look at the drive from MCI, mild cognitive impairment, to dementia. And UNC Pembroke is a wonderful school. Look on a map so you know where Pembroke is. So you can come to the slam dunk in May. That's where it'll be. And whether I'm talking to undergraduates or some high school students today, I don't know, or my favorite 3-year-olds scientist, Sophia. Let me explain that brain you have in your head. It has billions and billions of cells, up to 100 billion neurons, which are very specialized cells with very long wire-like projections. And in your brain, it's hard to fathom 100 billion. So I always tell people, think of your brain like the planet Earth. There's a little bit of a size difference, but every nerve cell is like a house, and there are just as many homes, condos, trailers, and offices on the planet, just like there is in your brain. There's a lot of similarities between our planet and our brain. There's oceans on the planet. Those are like the ventricle fluid-filled spaces of the brain. When you zoom in on the Earth, you'll see continents. Those are like the major regions, the frontal cortex, the cerebellum of the brain. When you start zooming in on the Earth, you see more detail, just like when you zoom in on the brain with specialized instruments. You'll see countries and cities, and eventually, looking at the Earth, you'll see neighborhoods, maybe the house you live on a city block, where nicely organized houses occur, just like the brain. The brain has neurons that are organized in neighborhoods, packed in just like homes in San Diego, where my parents lived, where they like to build homes very tight together and in nice little structures and layers. And that's how you can think of the brain. Just like all the cellular neighborhoods that are all talking to each other on the planet Earth with wires and telephone communications. And every cell has machinery, just like a house has a furnace, that's the mitochondria, has a library, that's the nucleus, etc. And cells take in nutrients to make replacement parts to keep the nerve cells working efficiently, just like you bring in nutrients to a house, except you have to use the nutrients to help you be nourished so that you can fix the windows that get broken or the appliances. So it's very similar. And if things go wrong, if something's not working properly to get rid of old and damaged equipment of a house, if you're bringing in groceries every single day for 10, 20, 30, 40 years, eventually the inside of your house might look like this. And if you look at neurons in Alzheimer's disease brains and look inside the neuron, it looks very similar to this picture. You might not see details of cracker boxes and beer bottles, but it's very similar of damaged pathogenic material accumulating. In my lab, we're interested in what's the wiring circuitries that become vulnerable during aging and other types of brain vulnerabilities. There's lots of work in the field that seizure events or traumatic injuries, even mild injuries that are not at the level of TBI can affect synapses and make the synapses then vulnerable, so that you're at a higher risk of dementia later in life. So all these neurons they're nicely organized in neighborhoods. When you zoom in on those individual nerve cells, you see they're very complicated by just how many connections they can make. A single neuron has up to 4,000, even more, places where other neurons can make a connection. And they use these little spine things that are like telephone receivers, where another neuron can send a wire and make a connection. And that's called the synapse. And if we zoom in on a single synapse, my lab is interested in all the orchestrated events and mechanisms necessary to allow synapses to allow neurons to communicate and for neurons to encode information, for memories to be stored. It's these synapses that determine your personality and the clothes you wear, etc. All your information and aspects of your memories are embedded here. And there's different components of the synapses that have vulnerabilities with stroke events, seizures, and traumatic injuries, all of which are known to increase the risk factor of dementia. So we've been trying to understand those type of connections, whether it's cytoskeletal changes, adhesion dynamics that Michael will go more into on one particular aspect. So we do all kinds of work. And we try to start most of our research in a three-dimensional culture model. These are slices of hippocampus, because the hippocampus is important for learning and memory. And that allows us to focus on our experimental parameters without worrying about the breathing rate of the animal or the circulatory system, et cetera. And we can look at the kind of risk factors, whether it's hyperexcitability, we can induce seizures, or age-related protein accumulation events, and then even TBIs that might be also involved in increasing dementia risk. And the brain responds with different types of signaling, both pathogenic and repair responses. And our work has been finding those and how they balance and how they interact. So you get the final culmination of what is happening to the brain. Sometimes you get recovery from minor seizures, but then sometimes you get gradual cell death because you've gone past a point where the repair responses can't counteract the pathogenic responses. And all of this work is trying to understand how other types of brain vulnerabilities influence age-related synaptic decline. We are all losing synapses as we age. Some of you younger folks, you may not have been out to the parking lot and stared at an empty space and said, someone stole my car, because you happen to park somewhere differently that day. It takes me sometimes a few minutes to say, I don't know why my car is not there. That's normal age-related synaptic loss. Alzheimer's is accelerated events of synaptic decline. And all these brain vulnerabilities we study seem to influence synapses and affects that type of event that increases the risk. And Michael's going to go through some details. We have lots of papers where we can keep these hippocampal explants in culture. They live on a little membrane, so they get oxygen and air from the air interface and nutrients from below. We put media underneath the little inserts, and we can maintain tissue alive. They're three-dimensional, about 15 to 20 neurons thick. And you can, with your naked eye, you can see CA1, CA3. It's a pretty cool process. You can put electrodes in them and measure the same electrical activity you're using in your brain right now. And we put on different types of toxins, whether it's pesticides that might be an issue if you live near a cornfield in Pembroke and they're spraying on certain weeks, or the Army will want us to put on low-dosed nerve agents like SOMAN, the very dangerous nerve agents, to see what low exposures do. We're not interested in just killing the tissue in seconds to minutes, because everybody knows about the toxicology of certain things. We want to know what happens when someone's exposed to something, their brain's influenced for a while, they could be dizzy for a day, the next day they seem fine, but what's happening to their brain a week later, a year later, or 4 decades later, where it might influence the onset of dementia later in life. And there's lots of data that we can study synaptic integrity. And we also are very interested in adhesion molecules and cytoskeletal components, because those have been linked to synaptic plasticity mechanisms. So if you're disrupting those, that can affect the memory encoding processes. So Michael will get more into those type of works. I'll take you through just a little way to look at how we monitor synaptic integrity. On a very simple view, this is just a time course where it takes us just a day to make the tissue, but it goes through a clearing phase. So we don't use the tissue for 2 to 3 weeks in culture. Because realize it's like a big loaf of bread and we throw a blade through it to make a slice of hippocampus. So the tissue is damaged and it has to recover from that. The dead neurons, damaged and dead neurons clear out. And after 2 or 3 weeks, we get a very healthy looking tissue. And then we can monitor pre and post-synaptic markers. There's many markers that are very good at establishing synaptic integrity, and it stays very constant. We've followed these cultures for months. Some people have done a year or more, but we typically do experiments for 2 to 3 months. At culture day 25, oftentimes when we start our experiments, we can put on different insults. These are canic acid and different kinds of excitotoxins. Peroxon is one of those pesticides. And we see this rapid decline in synaptic markers. So we call these rapid instigators of disrupting synaptic integrity. And using this type of culture system, we can see that other types of toxins do slower events, whether it's mitochondrial toxin, which is a slower process, or mimicking aging in a dish where we can use the drug called chloroquine, which will disrupt some of the protein clearing machinery that nerve cells use. And those protein clearance machineries are some of the age-related changes that our brains are dealing with when we get into our later years in life. And electrophysiologically, we can put electrodes in, showing that we're not looking at rapid changes. We're not looking at a toxin that immediately turns off a receptor channel or causes excitability. This is something where we see pretty much normal responses for 3 or 5 days, and then gradually you see loss of channel activity. And some of the work many years ago used this system to show that if you pre-treat with canic acid, which causes seizures, and this is just like a minor seizure that someone would have, that after the seizure, just like my dog would have, they're fine. They shake themselves off and they're okay, and they have those once or twice a month. And we showed that just very mild seizure response makes the secondary activity of an aging insult like chloroquine to be more rapid. And then the chloroquine toxicity is no longer recoverable after pre-treatment with canic acid. So these are the type of studies we've done to show that why are stroke patients more vulnerable to Alzheimer's and vice versa. We're showing those type of connections with doing pre-treatments of hyperexcitability. And then regarding TBI that Michael's going to go into, that whether it's sports injuries or military blasts, we don't do much of the physical TBI work. The Army came to us and wanted us to test some of their military explosives. They have experts that know how to make very precise and routinely reproducible plastic explosives. In this lower left panel, you see like a pea-size plastic explosives. And these are the experts at Aberdeen Proving Ground. It's one of the Army bases. And I don't want to be around when they're making those things, but they're able to allow scientists to use those, whether they're exposing PC12 cells in a dish, and they wanted us to bring cultured slices. So they asked us to drive our tissue from UNC Pembroke, that's kind of between the two Charlotte and Wilmington UNC campuses, and drive it all the way to Aberdeen Proving Ground. We had a little incubator in the back of the car every time we did this. And they would do the blast. We would keep them alive for a few days there, or drive them partway back and do electrophysiology in other people's labs. And that's what Michael's going to talk about. So I'm going to stop sharing, and Michael Almeida will take over. You there, Michael?
Yes, can you see my PowerPoint? Yes or not?
It's coming up.
I'm not seeing anything now.
Did
There you
you?
go. You see my first
There you
one?
go.
Yep.
Okay. Thank you all for having me again. It's my third consecutive presentation with ADRC. I'm so happy again to be with you all here. And today I will talk a little bit about my favorite project in BioLab. I've been working on that since 2016 is when I exactly started to work with this one. And that's not my PhD related project, but by far away is my favorite one. And I just keep it talking about what Dr. Barr introduced to you all, that is this synaptic pathology and the risk for dementia. As Dr. Barr showed, we have different models in the lab where we study the synaptic disruption and synaptic integrity disruption and how this can increase the risk for dementia. And also we have a couple avenues that we are testing and try to see how we can avoid the delay or stop it. Specifically today, I would like to talk about this traumatic brain injury. And I just put here one background of traumatic brain injury that is a force, external force, that cause some pathology in the brain, but not necessarily has symptoms. You can have this, a subtle TBI, that you can just have a concussion without any morphological change in the brain, or you can have a high level of this mechanical force that can make you hit your head against some wall, and that can cause physical damage. And there are many other examples, like if you are inside a car and have a car accident, the airbag inflates, that you're going to create a pressure in your core. That internal pressure is going to like change the pressure inside your brain, is another type of mechanical force, or that's pressure that's gonna change. But again, I don't want to get details about that, but just for you guys understand that what I'm talking about when I mention TBI. And also are different levels of TBI. I think mine is a little bit slowly. Okay. And I changed the slide, correct? And then is it in when specifically today I want to talk about blast, this explosion that induced TBI, and we have 4 levels. In this image here, we are just seeing 3 because the 4th one normally causes death, and then I don't want to discuss that today, but we have primary blast, where it's more like the injuries caused by the blast wave. Secondary is also blast wave, but something that hits your head. And third one, and you hit something against you, like you hit your head in the wall caused by that blast. But again, today in the model that we have is a primary model. We just have this, if you did the CDC definition and classification of that, when we have a high pressure blast, and this causes into the brain and to the nervous system some concussion without any physical signs of pathology. And where we experience, or where we can experience this type of shock wave that causes this primary blast that induce TBI in war zones, unfortunately. And I'm just put here one image about the most recent war that we are facing right now. Over there, we have millions of explosion, all then create this blast wave. We can have that in terrorism attack. I put here an image from the Boston Marathon terrorism attack in 2013. And then here is the part of Department of Defense, U.S. Department of Defense, sorry, report where they are measured different levels of this blast exposure by just shock waves, and how this will affect the individuals depend on the distance and how much pressure they has been exposed, just this air pressure exposing, how this can affect. They are studying that to create like some equipment to keep the soldiers healthy during those attacks, or also civilians, but that is just one example. And another one is not for all civilians, but for military training. When they are in training, not in war zone, they are like using these big guns. You can also see a blast wave behind them. And the same report, they also measure that and how these blast shock waves can different affect the brain. And I'm just put a note here, don't take me wrong, but I live in Fayetteville area for 5 years. And if you live around there, you're gonna list at least twice a year those blasts happen all night long. Normally it's Ray Marine are there, and that make me so confused because I'm studying blast shock waves caused by military explosion, and all the time then I heard that during the night, I said, oh my goodness, I hope someone is measuring that I'm low level of exposure to that. Now I'm not living Fayetteville anymore, but I'm still thinking about that every time when I go back to this study. Okay, but what I want to highlight here is that it's a different types of TBI, and we are not discuss the level of TBI. That's another discussion. But even if you have subtle exposure to TBI or low pressure TBI, you're going to increase later in life the risk for dementia. Here is just Alzheimer's Association code, where say a history of moderate TBI had a 2.3 times greater risk to develop Alzheimer. Military person, they have a 4 to 5 to 6 times great risk to develop Alzheimer. If you have just a single TBI, that don't have any evidence that you're going to develop any dementia late in life, but as you have a chronic exposure to that, you might have, and different than what's happening in sports like football, where you have more impact head to head in development the chronic traumatic encephalopathy, the CTE. TBI is more like in low dosage, but along the life, you can increase your risk for dementia. And how we study or how we try to understand the pathology behind these primary blasts. Again, I'm started to work on that in 2016, and this is just one example of the blast wave. And in the paper that we published in 2016, we showed that across the time after blast exposure, are you going to get details how we do that explosion in a moment? We have a progressive decline of some synaptic markers, and that's happened in absence of neurodegeneration, happen in absence of neuropathological damage to the cells. Here you can see in these two panels, the control, the 3x blast is like 3 repetition of these detonations, and a positive control for a neurodegeneration in this case. And then you can see we don't have any sign of neurodegeneration, but we have a markedly decline of these synaptic chains. And we do not see any signs of recovery up to 96 hours. I don't know if after that we're gonna have signs of the recovery, but up to 96 hours, we don't have any signs. Okay, but the pathology behind that, or the mechanism how these blast shock waves will induce this synaptic decline, can be multiple ways. We can have a cascade that can change the proteolysis event, the protein deposition, change the cytoskeletal, change the adhesion molecules all together, multiples of them, and that can lead to that progressive synaptic decline. And there is what share with risk of dementia, because if you have this TBI type event, seizure, this blast exposure, one example of what can happen is we can have a disruption of this glutamatergic signaling. By disruption, for example, the receptors of the glutamatergic signaling in the synapses. It's going to affect the synapse. It's going to maybe increase the influx of calcium. That's going to cause some protease activation, cytoskeletal breakdown, some changes in the structures, cause synaptic decline, and leads to neurodegeneration late in life. To give you guys another idea how this pathway is important, one of the few drugs approved for Alzheimer's disease is Namenda, that works exactly on this pathway to try to regulate this receptors activity, or this receptor binding activity, to try to to minimize the symptoms of Alzheimer and cognition problems related with that. Okay, but how we study that? We use this called brain experiments, the organotypic cell tissue culture, and we use a specific region in the brain that's called hippocampus. And if you have a brain, the hippocampus is going to be basically on this region. And I just flip here for be easy for us to understand. This is how a mature and native hippocampus from the human brain is going to look like. We have specifically regions where we can see cell bodies is these dark purple regions where we have cell bodies. On these light purple regions is where we have most of these synaptic connections happen. And our model, this interface organotypic hippocampal slice culture, has been well established in the field. This is a model that has been used to study neurodegenerative disease or neurodevelopment of the brain. These have 3 points that I always like to highlight that is really important for us to understand the synaptic integrity in the brain studies. First of them is a 3-dimensional organization of the brain. As Dr. Barr mentioned before, we have cells, 10 to 15 layers of neuronal cells. And if you think they are organized up each other and also side by side with each other, that is how our brain is organized. We have connections in all the 3 dimensions directions. The second one is a multi-cell type in our brain. If you remember my previous presentation, we don't have only neuronal cells, neurons. We have also glial cells. And on this type, we can keep both neurons and also glial cells like astrocytic cells or oligodendrocyte cells. If you is interested of study, we can keep those alive, microglia cells alive. And the third one is this is a versatile to translation studies, because this is an interface between a monolayer cell culture and an in vivo model. Couple research called this ex vivo model. We have a kind of easy or a less issued process for translation the results that we have in the tissue culture to the in vivo model. Okay, I sell for you all my model of organotypic slice culture, and we are happy to do collaborations. But here, I'm gonna highlight how we did this blast, how we created these blast shock waves to induce this type of changes in the brain. As Dr. Barr showed before, this is a collaboration with Aberdeen Proving Ground, the Army Research Laboratory. And here is the exactly picture about the apparatus that they built to do this for us. I will use this left panel here, on this right one, I don't know now, but to illustrate that, and you can see exactly what we have here is what I have on this side of the slide. This is a water tank, and then we put the cells or the plates with the tissues inside here. And then you have this 6-well plate, we have 3 pressure sensors above. We keep these 18 centimeters from the wall. This is a glass wall here. Is a kind of the distance of our hippocampus from our frontal head. And then the detonate this explosive, the RDX explosive is like 15 centimeter outside. Depending on the model, when we want to study high dosage, we move this closer or far away from the wall. But this is just for you guys to understand. And then on this image here, you guys can see the water tank filled with water, the plates inside there with the tissue, as Dr. Barr explained, organotypic brain slice. They have a mature and native organization of hippocampus cells. And here, this explosive is going to be exactly on this point here, 15 centimeters, and you're going to detonate. Here is a video from the Army Research Laboratory where they illustrate how the detonation happened. After they have the detonation here, you're going to create an air shock wave, going to hit the wall of this water tank, and then you're going to create a water shock wave inside, kind of reproduce what's happening in the real time. What we do, they are more engineers and biophysics, they are interested in other properties of that. But after we do that, they give us the amazing brain tissue for us analyze. Here is the paper that we published last year about that, and we follow up a couple synaptic markers to see if they will change differently or equally other insults when they are exposed to some level of this blast induced TBI. And then we can see here, most of them we have a reduction, but a couple of them we don't have. And that leads us to understand that we have a kind of a distinct synaptic disruption for blast-induced shockwave. When we compare with other models that we have in the lab, is not the same. It's kind of blast cause a signature, or blast, sorry, has a signature of this synaptic decline, as we can see, at least on this graph here. And also we did some immunostain and loop confocal image to see this reduction. We can see on the top image, they are brighter than the bottom image, what means we are losing density, we are losing positive marker of this synaptic marker. Okay. Next, as Dr. Barr mentioned, a couple times we stop in the middle of the road at Research Triangle Park to do some collaboration, and we handle those tissues to Serena Dudek at the National Institute of Environmental Health and Science to do some electrophysiology analysis. And as an outcome from that image, we have subtle change on the plasticity. That means the tissue exposed to RDX blast do not like lost his ability to have a actual potential, do not lose his functional ability, but change a little bit. And one of the most interesting result is they kind of get hyper excitability. It's like it's hard to then to get back to the control condition, or to like the rest condition. It's harder to then to go back to the normal condition after they have the blast exposure. And that's kind of represents some type of others insults like epilepsy or others type of disease or brain issues that can increase the risk for disease. I don't want to get in detail about the neuroplasticity and how that's done, but if someone want to talk about that, we can chat a little bit more. But just to show here, we have a structural, because I show you guys some changes in specific synaptic markers, a distinct change caused by RDX blast, and also we have a subtle change on the plasticity that is the functional of these synapses and how this can affect the brain cognition. And also in the same study, we find some markers that can be used as a biomarkers to identify this level of disruption in the brain. And again, this is important to understand. We don't have any symptoms. This level of TBI don't cause any symptoms. We cannot see, like, if this is called a subconcussion level of TBI. But one of these biomarkers that we found is called NCAN, is one of these adhesion molecules. And NCAN have different isoforms. And we can see specifically here, after RDX exposure, we have a higher reduction of the isoform 180 kilodaltons, as we can, the bar, the quantification just show us here. But besides this reduction on this specific isoform, what we found is, after RDX exposure, we have some breakdown products of this NCAN that we don't have an under control condition, and that might be potential antibodies targets to be used as a biomarkers. We have specifically two. One breakdown product around 70 kilodaltons and another one around 35 kilodaltons. We also did some immunostain. I will talk about tau that is in the bottom panel here in the next slide. But here we also confirm that, yes, along with the synaptic and functional change that RDX causes, this also causes some change on these adhesion molecules. And specifically for the NCAN, the blast exposure appears to produce a breakdown product specifically distinct for this type of insult that we don't have in other insults. And just to recapitulate, as Dr. Barr said before, we are monitoring this synaptic integrity by different models of insults. We have these excitability models. We have this chloroquine that I mentioned in my previous presentation. We have this Alzheimer-type protein accumulation stress that also cause a synaptic integrity, but a slowly one compare. And we have the blast one that appears to be not fast in time, but a bit appears to be happy to compare with others. Okay, and what else we can see or we found when we look for this synaptic integrity? synaptic integrity change caused by blast exposure. We look for one of the marker, the GFAP, is a marker for astrocytic cell that has been listed as a biomarker for TBI that blast that induce TBI. And here on those panels, we have a confocal image. In the green, we see synaptic marker, and here's the graphic for that. We can see across this exposure to RDX, we have a progressive decline of this synaptic marker. But we do not see an overall change in these astrocytic positive cells labeling with GFAP. As we can see, we have a trend of increase, but this do not represent a significant change. Okay, but the literature has been described this as a biomarker for blast-induced TBI. Then we have wonderful undergrad students here, and a couple of them help us to do this analysis. We have a kind of high resolution microscopy here, and we are looking in an overall, on a broad picture. We don't have change on these astrocytic cells. But when we did cell analysis or cell morphology analysis for a single cell, we start to find where is those difference. And just to exemplify here, we analyze the cell body size and the number of ramifications across multiple cells inside those tissue, exposed or not with that blast. And we find two interesting responses. The first one is, after blast exposure, those cells tend to have a bigger cell body. They kind of get bigger. And if we just follow across the time, not in the initial moment, the arms tend to be shorter. Is another researcher that also studied this. He called this as a clamastodendrosis. It's like a apoptotic process of death of these astrocytic cells. I'm not calling this here now, but I'm just telling you, we see at this lower level of a blast exposure, we are seeing some change in the morphology of this astrocyte. But remember, this is not enough yet to show change in an overall picture. I don't know if that is because the time, if I analyze that later on, like weeks later, if I'm gonna see change, or it's because the dosage, I don't know. We are keeping looking for that. And another interesting point is just the blast exposure tissue produces this this GFAP, this same marker breakdown product, as we can see here. And we have GFAP standing for a bunch of different models, and we never saw this breakdown product. And again, can be another target for biomarkers, antibody labeled biomarkers. Okay. What is the next step of this? We are trying this collaboration. We are like in the process to exchange samples with Daniel Pear for Uniform Service University. This is a kind of DOD institute. And to try to analyze if we have the same change in the human brains from veterans. They have a bank of brains over there of post-mortem brains. If a history of blast exposure and those TBI events. And is a very interesting because it's a high level militaries and they have a history of pressure for how long, for how many times those brain was exposed to this military blast wave. And it's going to be very interesting what we can find from that. But again, that is just initial. We just start that like last year. Okay, what else we found? The next one is very common. We found in literature that blast-induced TBI is gonna cause a tau pathology. And then what we did, we analyzed these pretangles in our model. And here is interesting, just because just when we have high level, let me put that in other words. We study subconcussion levels, but inside that subconcussion levels, we have different levels, lower to high. Inside this high level at under subconcussion level, we have positive marker of this pretangle of tau that is can maybe be a sign of tau pathology. If you see, when we have a lower level of exposure, we do not have this positive marker of pretangles of tau. And also we have a inverted correlation in the levels of synaptic marker and the presence of this pre-tangle of tau. What might indicate, yes, we have some type of tau pathology associated with this synaptic decline. Also, we have some others marker like ubiquitin here that is just shows some problem others behind the synaptic decline on these cells. Also, another step that we are taking right now, because we are kind of start to see this synaptopathology, is one other or another marker for biomarker for this blast-induced TBI is axonal damage, and it's a different levels of axonal damage, different models to study that. But as we can see here, Daniel Pear, he just published a paper also last year about this topic, how we can use axonal health, the neuronal axonal health, to tracking this TBI, blast-induced TBI. We are just started to doing that, and that make us make a new collaboration with like a microscopist person who have like fancy microscopy to do better analysis than what we can do here. But we just start to see what level or how is the health of the axonals after blast exposure in different levels of exposure, different numbers of repetitions, and everything else. Okay, just to summarize what we study on this project is, we have this unique military blast because they definitely use C4 to cause this explosion. And then we have two types of response here. We have one response that is without any symptoms. We cannot have like concussion with this level of blast, but we can see a distinct synaptopathology, a disruption in synaptic plasticity, changes in adhesion molecules, and subtle changes in the morphology of astrocytic cells. This might be one of the mechanisms that cause alterations in behavior, anxiety, depression on those people exposed to these lower levels of military or any type of blast. In another way, we have some markers that are leading us to understand better how these blast-induced TBI increase the risk for dementia, because we have this pre-tangle accumulation of this tau protein. We might have some axonal injury, and we have these changes in inflammatory pathways like GFAP breakdown products. And then those can maybe help us to understand the pathology, but also a couple of them can be used as a biomarker of the disease. Okay, I'm almost done. Here is to say thank you for all the colleagues and collaborators that help us in this study and all the fund agents to do that. And now is the most important slide. That is, I hope after 3 consecutive meetings, you know how to say thank you in Portuguese. If all that, I will take any question. Thank you so much.
So this may be a really ignorant question. And it's, is there any male-female variation in the hippocampal samples, or is it gender neutral? That's a real back story, but I'm just curious about it, sort of as you're generating those slices.
I will be honest with you, I really don't know, but I will use our model to say, when we do this cut section, we have the pups with 12, 13 days old. We don't know if they're male or females, or we did not try to identify that. And then they are separated randomically inside those samples. And after that, again, I cannot identify if that came from a female or from a male hippocampus. Maybe it's you're going to be interesting if you in some point we do that, but I don't know if it's any difference on that. And on this data, we don't kind of do not separate that, discriminate this problem because they are mixed together. But please, Dr. Bar, help me to answer it.
The male-female question is a big topic, but not in our lab. I
Okay.
follow Gary Lynch and Christine Gall from UC Irvine. I did my postdoc work there, and they just published an interesting paper. It's not on TBI, but showing differences in hippocampal circuitries at certain stages of males versus females, and they have a few other papers, I think, in their repertoire. So there are some interesting things. Of course, once you take the brain out and they're in a dish, I don't think we would see changes, but they're doing the in vivo work. It's very cool stuff. Any other questions, everybody?
Have you thought about looking at, you know, I mean, I know you're interested in looking at some of the underlying sort of mechanisms that do this. I didn't notice if you have been or are interested in doing any like epigenetic evaluation in these samples, maybe, you know, that either have or haven't been treated with blast exposure. It's, you know, a possible, I guess, mechanism. There's plenty to sort of explain some physiologic differences and what might be happening in response to the blast to mediate these effects.
I can say that we had lots of ideas we wanted to chase. We ran into trouble that the Army doesn't make a lot of these blast things, and they have to have their internal money. It doesn't matter how much grant money I bring to them. They have to have internal money for the blast place where we take them. But we've had plenty of work we wanted to do, and there's even work of exposing the animals themselves to blasts and then taking the tissue out and making cultures and see what happens when we do very distinct changes in the culture setup.
We had a comment in the chat from Scott, which is one of the reasons I had reached out to invite him. So well, I should maybe just let him say it, but he did put his contact info there for you both.
Yeah, just not to take too much time, but we have a short-term model of brain slice. It's kind of a whole amount. It's not just hippocampus. We kind of look at other stuff too, use it as a brain tumor model a lot, but we have some interesting constructs with like mutant tau that we can transfect in and cause neurodegeneration. So it's something we might want to compare notes, because I really love some of your image analysis, some of the things that we could really learn a lot from you. That'd be great.
That's all Michael's doing. He's set up our nice microscopes, and we got pushed in that nice direction of imaging with people at Chapel Hill, Vladimir, who's now at CZI. So we're trying to push that type of work. And we've had other potential collaborators. I forget the name of the school, Michael, where they wanted to take cadaver. No, the other one in North Carolina. They wanted to take a cadaver head. Yeah, High Point, a cadaver head. So we would have the exact physical components of a human skull and even the brain. And then we would put a plate of tissue in there, but I really didn't want to be involved in that. But it's still up in the air as far as doing that kind of work too.
Let me just add one comment to that about your chat, and thank you for contacting us. But we kind of have the same idea. We work in collaboration with Todd Cohen sometimes on that. This is most frozen right now. Again, it's not my PhD project, but we get like a side project on that. And one idea that we have before that we try it is if we can if these brain slides can uptake human tau. And I tried that, and yes, because Todd Cohen kind of wants to separate humans, different tubes of tau, give to us, and we put on this, see if the brain can uptake that, and then submit them to the blast and see what's happened. And we are trying to expand this a little bit, not only hippocampus, but have can you help me, Dr. Bar? entorhinal cortex.
I'm
Yeah,
gonna go-
entorhinal cortex, and see, because Karen
Yeah.
Duff said about tau spread out, how this happened, and then we are kind of connected at this point. But it's gonna be great to chat a little bit more about how you can study that, and if you are expertise in what we do here. But thank you for the comments here.
Great.
And the push here from the Army and NASA has funds for some of our undergraduate work is how does the older brain respond to these type of things, whether they're in space travel and there's turbulence and shock waves, or militaries, that everyone seems to be working longer. I see people in stores that are 70s and 80s working, and we're going to have astronauts that, yes, we've had someone in space who was 90 years old. And so now we're worried about what vulnerabilities the aged brain has when it comes to all these factors. So we'd like to start doing animals of the different ages and doing this type of work.
I think we have Dr. Z have a question.
Yeah. Hi, Michael. This was such an interesting talk. I heard Ben's talk like some half a year ago when you were at ECU. And since then, you really progressed a lot with this, moving from animals to human models. So I like very much this collaboration you were talking about, to looking at these astrocytes with enlarged somas in slices from deceased veterans. And I like that because there you can look at sex, at age, brain region, right? You can kind of connect to the type of blast and time, and you can have lots of multifactorial analysis. So I was wondering if you could just take out with a laser capture dissection microscope these astrocytes from different brain areas and run transcriptomics or proteomics on them and see if these are protoplasmic or fibrous astrocytes. If the blast affects, I assume must be protoplasmic, right? Because soma.
Yes. Oh, that is a really interesting point. My wife, she's gonna be in one of the talks some point here about the other project that we have, I think November. And she studies specific astrocytes. She's who help us with those analyses that we have here. And the first thing is the military samples from human brain. That's very interesting. Is most interesting because we can test the chains if we can translate what we found in our model in humans tissue. First one. Second one is can be identified by location because we just look in hippocampus, but we can see across other brain regions if we have that difference. And third one is because this is a kind of military related project that they have, they do not, the difficult for us is like get the samples because it's hard, this like paperwork process. They have a really good background history on the TBI. The pressure has something called impulse, that is the pressure level by the time from those human brains. And they have everything. This is a high level militaries and they track everything from them. They have blood samples, they have everything. What is pretty good, if we kind of can find the same, is gonna be a pretty helpful data. That's the first one. About the identification of which type of astrocytic cell is that. I wish I can call to my wife. She will know how to answer that because she tried to teach me. I didn't learn it yet, but hippocampus trained to be just the photoplasmatic, something like that, the name, yeah, because it's the type of the tissue, the features of the tissue. Don't means that you don't have others, but most, like 90% probably is gonna be this type. The interesting point of your question is maybe those subtle changes just happen on this 10% of the others type, and that is why we cannot see in the overall picture. Is a, again, I never thought like that, but it's a very interesting point how we can analyze. Now I need to call my wife and discuss how we can analyze that. But thank you so much for the question.
Yeah, because with the microglia, we saw that microglia is bad in Alzheimer's or good in Alzheimer's with this discussion. And now, if I understand correctly, microglia in SLM and microglia in stratum radiatum behave completely differently. I think those in stratum radiatum are the bad microglia in Alzheimer's. So I was wondering if for astrocytes is the same, that these astrocytes with enlarged soma are actually localized in a certain area.
Yes, is again very interesting point. We did kind of analyze in different regions, like we have some stratum radiatum, we have some molecular layers, we have inside dental jar the hylux area. They do not trend to be like significantly different in the response from others, but again, is your point is a very interesting, and we should pay more attention and give more work on that for sure.
Yeah, you should come to our neuroscience symposium. I'm going to send to all of you the flyer.
Okay, we will.
You should present to our community because it's really, really cool.
Yeah, we just sent an offer letter out to someone. Hopefully they'll come. I think they will, to Duke. They're at City of Hope, and they do PBMC derived neurosphere organoid models, which other groups can do as well. But the thing that I've been impressed with their models, again, they're PBMC derived or fibroblast derived. They retain microglia, and the microglia continue to be there like 4 months out. And so we're enthusiastic about those as a model for doing all sorts of different exposures on, because we've talked about doing like toxic compound exposures with our Nicholas School of the Environment collaborator here, and looking at like epigenetic changes. But if you can have models with actual functional microglia, then you can flow sort and RNA-seq and do all sorts of cool stuff. So I will. I think they're coming out for another visit, and so if they're giving another talk via Zoom and you're interested, I'd be happy to send that hyperlink to Michael and Ben. And Erzsébet too.
Please do that. I think that is like the future is going to that 3D modeling,
3D
and
model,
then
yeah.
and again, Dr. Barr is on that, using that field since he's starting science, this model. And the big difference now is that they are reconstructing that for a human cells, like the fibroblast. What in our model we use rat cells or mice cells to do that, but I think the field is going to move towards that. We have others. I kind of love these small machines behind me, and this small machine now, the big challenge to them is how we can image further with a high resolution and also deeper on these 3D models, because we can do this pretty well in cells, but we cannot image pretty well like live cell image in tissue yet. And I think that is the two main points of the field where it needs to development, how you can analyze them, but also how you can have them more representative to the reality.
Yeah. Well, and just to get at questions like Tipa asked about, differences by sex. I mean, if you can make models from PBMCs, you can just get PBMCs from 3 men and 3 women, right? Or and this you can never do in animal models. So if you want to ask questions about like racial and ethnic disparities and differences, right? You just get PBMCs from, people from different racial, ethnic, ancestral backgrounds and try those models. So yeah, it's a very cool new future, and I'm excited you guys are so engaged with it. So well, we are at time. I want to thank you both so much for your talk. It's very interesting. Really appreciate it.
We're glad to help out.
Absolutely.
Thank you so much.
All right. See you guys.
Take care all.
Thanks, Michael.